Hello, welcome to our online poster session. My name is Joaquin Reyna, and I'm presenting with curated. And today we'll be presenting the loop catalog. To motivate this, I'd like you to think about genes and their regulatory elements. These can work through a variety of functions, but one of the most important is 3D interactions. In order to capture 3D interactions, we use the HiChIP protocol. However, it tends to capture a comprehensive set of interactions that sometimes might even cost too much. So in order to target our interactions to potentially just regulatory interactions, we can use HiChIP, which introduces an immunoprecipitation step, allowing you to target DNA-bound proteins such as histone modifications or transcription factors. So in effect, HiChIP is a very useful tool to analyze 3D interactions in a very protein-centric way. So HiChIP is actually derived from HiChIP with the addition of this immunoprecipitation step right here. And our overall goal is to collect a large collection of HiChIP samples to build a database of high confidence loop calls. We've gathered data from 2016 to August 2022, and we've seen that this dataset is actually exploding in the number of publications. In addition, we've been able to find 680 samples for which there's a variety of different pull downs, as you can see here. We've built a pipeline that's able to handle HT38 and T2T, as well as MM10. In addition, we call loops with three different strategies, one using just HiChIP data itself, another using ChIP-seq, and another using HiCups, a standard protocol that's used in the original HiC dataset, the data type. From there, we took all of this data, incorporated it into a database, and introduced new novel features for our website. We provide this landing page where you can select the reference genome and our three different analyses. Starting with a main page, our loop page, you can see here that we provide a WashU embedding where you can select samples and visualize the different loops that we called. In addition, you can filter for different metadata and download the raw data or WashU JSONs, which can be used outside of our website. In addition, you can click on one of the samples where we'll link you to the different metadata as well as a summary of the loops. And we also provide the community detection analysis where the loops are connected together into a graph, and we're able to find communities where genes can be interacting with multiple enhancers or genes with other genes, and so on. From there, we wanted to provide a SNP to gene linking analysis where we took HiChIP samples and intersected them with GWAS studies. In particular, we looked at 4 different immunological diseases and make them available on this page here. You can query them, and that results in a very useful page where we visualize the different samples, their loop tracks, and the relevant find map GWAS SNPs. We also provide this table with useful buttons that let you navigate between the different SGL elements. To get a further idea of what we actually found with these SGLs, we did a summary to find out how well do we actually prioritize the GWAS SNPs, and we can see here on the top left that we start with a significant amount of SNPs and reduce them quite a lot, and that's the same across all these diseases. We found a variety of genes. And we wanted to delve into T1D. We built a consensus gene list where we were able to find approximately 500 genes and we overlapped that with our 643 loop catalog genes for T1D. 64 were overlapping and 580 are novel and unique. So in addition, we were able to find that most genes are connected to one SNP and one SNP to one gene. So this is very useful because this one-to-one will make it really easy to analyze. In addition, we also looked at this Mori et al. 2022 study where they showed that RS7292838 is actually influencing bacterial expression in T cells. We actually see this in our SGLs for CD4 T cells and CD8 T cells, but not for other immune cell types.
As a second application of our database, we performed a series of motif analyses in loop anchors, specifically regulatory interactions. We defined 2 high-confidence H3K27AC HiChIP sample sets, the A+ sample set composed of 5 immune cell types and the B+ sample set composed of diverse cell types. For both sample sets, we identified conserved loop anchors occurring in 90% or more samples. We performed motif enrichment analysis on these anchor regions using meme suite C and discovered ZNF460, 135, and MEF2A to be top significantly enriched motifs. We additionally confirmed our finding of ZNF460's significant enrichment through de novo motif analysis. With the motivation that DNA binding proteins such as CTCF are commonly found in a pairwise fashion at loop anchors, we performed 2D paired motif enrichment for these sets of regulatory loops using meme suite VMO to first annotate anchors with the Jaspard 2022 core motifs, and then used Bootstrap to identify significantly enriched pairs. ZNF460 pairs were among the top enriched pairs. However, as ZNF460 is a crab zinc finger protein, the crab domain being well established as a repressive domain, we found it surprising to see such strong enrichment of ZNF460 in H3K27AC HiChIP loop anchors. To further confirm our findings, we collected external ZNF460 ChIP-seq in HepG2 and 293 T cell lines and examined the overlap of CTCF, H3K27AC, and H3K9MA3 ChIP-seq signal with ZNF460 signal. At 2 loci in HepG2 cells, we indeed see overlap between ZNF460 and H3K27AC signal, as well as CTCF. To quantify this overlap genome-wide, we looked at the coverage of transcription factor and histone ChIP-seq reads across ZNF460 peak regions and observed the preferential binding of CTCF to zinc finger 460 peak sites, and similarly for H3K27AC, but to a lesser extent. Taken all together, we hypothesize that zinc finger 460 plays a dual active and repressive role in gene regulation through chromatin looping, and we plan to continue further investigation in the future. In conclusion, here we present Loop Catalog, a catalog of uniformly and expertly processed loops derived from publicly released HiChIP datasets. We aim to continue to expand Loop Catalog with additional tools such as differential loop calling, as well as recently released datasets. We'd like to acknowledge the following people for their contribution and support of the Loop Catalog project. Thank you for listening.